So welcome, happy Halloween. It's really nice to see many familiar faces in the audience. I've been sort of given a daunting task to cover sort of the breadth of how modified oligos are used. And so I'm really I'm the only speaker for this whole thing. So it's really 5 talks. And I'm going to start off with oligosynthesis, possible modifications and their use, talk 1, then molecular diagnostic basics, because that's what this whole meeting is about. I'm going to give you examples of how we sort of have innovated using modified oligonucleotides in a couple of areas, the HPA assay, something called switch blockers. I'm going to put in a little vignette about therapeutics. Then I'm going to talk about messenger RNA synthesis to enable protein synthesis at your will, and then wrap things up. So, other themes of my presentation are going to be the what, why, and how. So this is going to be a very different talk from what you've been experiencing today. The one thing in common, however, that I can guarantee you every other talk you've attended uses oligos and modified oligonucleotides. So it's my endeavor to make this talk one where I help with the how, tell you more about modified oligos, and then the why and the what for you to invent, create, enable whatever kind of solutions you want. In some ways, what I'm talking about is going to be a little bit of a walk down memory lane, because I've had the great benefit of living through the entire generation, starting at the beginning of molecular diagnostics. But what you're going to see is what is sort of looks old is actually new again. The first assays that we ever developed at GenProbe for infectious disease are still used today, still sold today. Along the way, I'm going to have various vignettes of where insight into seeing unexpected results. So expect unexpected, and when you see that unexpected result, explore it further, because it could lead to some key innovations and inventions. So just by way of history, we benefit by standing on the shoulders of others. Oligosynthesis started in the 50s with Todd, 60s with Khurana. Khurana used a difficult chemistry to actually elucidate the genetic code for what he got the Nobel Prize for in 1968. That further led to Bob Letzinger, Colin Reese, doing further development work on triesters, and then early phosphite chemistry, initially phosphite chloride chemistry, where coupling reactions went much more efficiently. But I'll tell you, having cut my teeth on a chloride chemistry, it was really tough. Fortunately, one of Marv Carruthers' students, Bob Letzinger's students, I should say, Marv Carruthers, came up with the amidite chemistry, which we still use today. So this is 1981. All of you in the audience benefit from all the intellectual property, all the work done with this common building block of phosphoramidites. And that additionally, much of that intellectual property was laid down in the early 80s to 2000 timeframe, issued patents, virtually all of them are now expired, and accessible for public use by all you guys to use how you like. I won't go into this in great detail. It's just the common cycle for phosphoramidite synthesis. But what I want to focus on is what kinds of modifications are available. And you very much benefit from the fact that there's all kinds of modifications that you can make to oligonucleotides without interfering with hybridization. So what I benefited from dialogue with a guy by the name of David Kearns in the mid-'80s. We brought him in as a consultant at GenProbe. He was a theoretical physicist studying duplex DNA. And what I learned from him was It's amazing what you can do to an oligo, as long as you aren't interfering with the sequences of bases, interfering with hydrophobic stacking, or interfering with Watson-Crick base pairing. You can do almost anything, and we do. It's kind of like almost a walk down the wild side in terms of what we can do. So here are some examples. We can completely replace the phosphate backbone with the likes of morpholinos or PNAs. You can do substitutions at the 2 position. You can substitute into the phosphate backbone, add linkers onto the 3' ends and 5' ends. All kinds of things that we can do now. Additionally, for attaching molecules to oligonucleotides, one of the first things that we did at GenProbe was come up with a way to make amidite synthons to insert directly into the backbone. Initially, we did those insertions as 3-carbon atom backbones, so that we're replacing one of the sugar bases, so we lost a base pair. But we rapidly discovered that we could simply insert that a 2-carbon atom backbone in a phosphate. We could do it with 3 too, but 2 worked better. Into the phosphate backbone, had very little negative effect on hybridization. Lost about 2 degrees in TM, but as you're going to see when we did the hybridization protection assay, we got it back when we attached things like acridine esters. Getting a little feedback. All kinds of other modifications that you can do here. In terms of base modifications, the first company I was involved with, Molecular Biosystems, a 3rd biotech company in San Diego. Jerry Ruth, that we brought in to be part of the team, did alloamine substitution on the 5 position. He went on to substitute all kinds of places. These substitutions are all ones that TriLink offers to allow you to attach various labels of your wish to nucleotide bases. Should mention that Jerry Ruth in 1996 through Molecular Biosystems got an issued patent attaching linker arms to any sterically tolerant site on a base. 1996, that patent's now expired. It's a freebie for everybody. These are some other examples that TriLink offers. At last count, I got 67 different dye conjugates that they can provide, either as directly or as post-conjugation, 10 different quenchers, and all kinds of non-fluorescent linkers. Here's an example of a biotin linker. So a little bit of a laundry list now of how these are used. So attach labels, you can insert bridging linkers. I'll get into that more with how we evolved to have the hybridization protection assay. Increase affinity, things like 2' fluoro, again, now off patent. ISIS, now Ionics, had the IP on that, but it's free now. And 2'-O-methyl, LNAs, methylphosphonates, ZNAs, TINA, on and on. Methods to increase affinity. Also a number of modifications to increase nuclease resistance, especially 2'-O-methyl. Phosphorothioates are used very routinely. Also block the 3' end, so it's not a primer. You stick almost anything on the 3' end that can't get clipped off. A phosphate, a propyl C3 spacer, a dideoxy. Can only do those by kind of the reverse direction synthesis, inverted bases, and lots of others. And then with the new series of phosphoramidites being available, that not only can we synthesize 3' phosphoramidites, it's now also available 5' phosphoramidites, so you can actually, in a single oligo, reverse direction if you choose to. So you want to have an oligo that has a loop in the middle and 2 3' ends, you can make it. And I've been making some of these for weird primers and things like that. We also have some modifications like ISO-C, ISO-G that will only base pair with themselves, so it can give you unique recognition. It's also available more on a limited basis, some left-handed helices. We use these at GenProbe to improve specific target capture. In the antisense arena, there's steric blockers, there's methylphosphonates, morpholinos, LNAs, PNAs. There's cleavers that utilize endogenous RNase H activity so that when the oligos hybridize, it cleaves with RNase H activity the RNA target. Those are called gapmers. And then additionally, synthetic RNAs, I'll talk about that at the end, and gene editing using CRISPR, using other kinds of oligos and oligo analogs. So a little quick about that's talk one. Now on to talk two. It's the basics of molecular diagnostics. And first, amplification. There's a boatload of these. It's becoming almost numbing how many different amplification systems there are, but probably everybody in the audience has used PCR at one time or another, or you have a friend that's used PCR. Transcript-mediated amplification, reverse or real-time transcript-mediated amplification. Those are both technologies used at GenProbe, strand displacement, rolling circle, LAMP, and many others. So lots of amplification methods to choose from. PCR, off patent. TMA, off patent. Strand displacement, off patent. I believe rolling circle is as well. So a plethora of things that you can get for free. Detection methods, radioactive, we've much gone away from that. Non-radioactive, that's what's rules today with chemiluminescent, fluorescent, and indirect labeling. These are some examples. TACMAN, there's probably hundreds of thousands of TACMAN probes that have been made now that use a fluorogenic label on the 5' end where the exonuclease activity, the polymerase eats its way in, cleaves that fluorophore off, and then you get a fluorescent event because the fluorophore has been removed away from the vicinity of the quencher. These are self-reporting probe and primer types. These are molecular beacons. Fred Kramer and his team came up with those. Then there's double-stranded probes where upon hybridization to a target, a quencher is displaced so that you get a fluorescent event. QPNA probes, where again, a fluorescer quencher upon displacement gets displaced, lights up. Another probe type, Vic Becker at GenProbe came up with this. It's very similar to a molecular beacon, except that molecular beacons, this whole loop region hybridizes a target, where molecular torches hybridizes a portion of the stem, a portion of the loop and into the stem, but in principle works kind of the same way. Then Amplifluor, again, a priming event and a priming event, a repriming, and double strand opens this up. Somewhat related priming event with scorpions. That upon extension, this sequence is also downstream here, so it will open up and hybridize downstream, moving quencher and fluorophore apart from each other. Then Plexor, where again, hybridization to a target displaces this. This is one of these strand separation ones. And then the black hole quencher plus probes upon hybridization. These are kind of folded up in quenching. When it hybridizes, it unquenches. This is, again, a similar one where hybridization to a target displaces that probe. This is used by Abbott in many of its assays. This is a probe I'll talk more about, called switch blockers. Talk more about it a little later in my presentation. That will be like presentation 4. And these both report there's a quencher and a fluorophore, so upon amplification, this construct will hybridize, separates the fluorophore and the quencher, but it's got a switch built into it, and that switch is designed using a thermodynamic trick to block wild type while not blocking mutant. I'll get more into how it works. So I'm now going to do a little deeper dive, and this is really coming to this sort of question of, what, why, and how. So GenProbe in the beginning, what we wanted to do is come up with super-sensitive assays that replace radioactivity. Why? So we could detect infectious diseases without people having to have radiation safety licenses. And I'm going to take a little dive under the hood. This is what you're not hearing at these talks, how this stuff actually works. So with actinium esters, as already indicated, is one of the most sensitive chemiluminescent labels known. Sensitivity is into about the 10 to the minus 3 mole, it's 10 to the minus 17 moles. And what lights it off is this actinium ester, like many chemiluminescent pathways, in the presence of a hydrogen peroxide and an alkaline condition, goes into a cyclooxetane. That cyclooxetane homolytically cleaves to give the excited acridone and CO2 and light. Firefly works like this, firefly luciferase and luciferin, cypridina, and many chemiluminescent systems. The bottom line is that if you hydrolyze that ester, there's no light because you have to have that ester for the cyclooxetane formation to get a light reaction. So we undertook to put actinium esters on probes, and this now brings up this aspect of getting an unexpected result, because Woodhead and Weeks for many years had been labeling proteins with actinium esters and making separation reactions and getting chemiluminescent outputs. We put an actinium ester on an oligonucleotide, and all of a sudden it was really unstable, far less stable than a free probe. So this is, again, this aspect of when you see the unexpected, explore it. You may figure something out that really can be important. So we did explore it. One of the first things we did was to synthesize oligonucleotide segments here that were poly A, poly T, poly G, and poly C. They were all highly unstable except for poly T. So the A, G, C all had exocyclic amines, and the T didn't. So surmised in part, what was happening is that those exocyclic amines were creating a general base hydrolysis and killing the actinium ester. It's about the same time I was diving into the work that Claude Helene had done with acridines. They weren't methyl acridines, they were acridines, but he was using them as intercalators, trying to use them for antisense therapeutics. And so with seeing this result of the instability of the free unhybridized acridinium, and then with the idea that perhaps we could put the actinium ester internal to the oligo, when it hybridized, the process of Watson-Crick formation would cap off all those exocyclic amines, and at the same time, the duplex formation would give a hydrophobic region that the actinium ester could interact with and exclude water to protect it from hydrolysis. So that turned out to work. So we had things that were really unstable, but when they hybridized, they became really stable. So had the benefit that one of the guys that joined the team, Mark Reynolds, the first project he started working on was a non-nucleotide linker to replace a base. So this was, again, a 3-carbon atom spacer to replace a base. And the first one made was called MSL, Mark Synthetic Linker. And so we took that linker, stuck an acridinium on it, and the hybridization protection assay worked very well. Here's, again, how it's working. So it's put the acronym ester on. For the free probe, unhybridized, that ester is exposed to the exocyclamines and that that hydrolysis environment. So very unstable. Clip the ester, and there's no light because there's no ester, there's no cyclooxetane. But upon hybridization, the acronym ester is stabilized in a one-to-one complex with the amount of target present. So we could drive these reactions with excess acronym ester probes to drive hybridization to completion and then do light off. But before that, what the hydrolysis rates look like of hybridized and not hybridized. This is a log plot. I had to put on a log plot so that one's not a vertical straight line, the other one is a flat line. It's a 30-fold difference in these hydrolysis rates, hybridized and unhybridized. So just imagine this, if you go through 2/3 of a half-life, you've still got this one, the hybridized, about 70% intact. 2/3 of a half-life is 20 half-lives of this one. 20 half-lives is a millionfold. So this gave us a way to homogeneously, just by hybridize at low pH where the acronym ester is reasonably stable, do it in excess to the amount of target you think is going to be there, kill the free probe, and then detect what's left. This is what the plots look like. So this was the homogeneous assay, and this was done in combination with ribosomal RNA. One of the founders of GenProbe in the very beginning was Dave Cohn. What he brought principally to GenProbe was the idea of using ribosomal RNA as a target. Turned out there was enough sequence complexity in ribosomal RNA, you could distinguish individual species with it. And the other benefit, because this is now the mid to later 80s, there's no PCR. There's no TMA. There was no reverse TMA. There was no rolling circle. So we used the natural amplification, 10,000 in a single bacteria. There's 100,000 in a eukaryotic cell, but prokaryotic's about 10,000. So we could take that acronym ester at about 10 to the minus 17, get up to about 10 to the minus 21 by targeting ribosomal RNA, so we could detect a few thousand organisms homogeneously in a single assay. So it was fully homogeneous. So this is old stuff, but it's actually new again because they're still used in assays. So it's fully homogeneous, 2 steps, less than 30 minutes, DNA and RNA targets, quantitative, linear over 3 to 4 logs. We also can detect single mismatches with it, and it's about 1 million times more sensitive than antibody assays. So layer into this. So this is now an onion story. You start off with the single core of the onion, you start putting layers on to make further improvements. As time goes on, we want to detect a lot more than bacteria with ribosomal RNA. We want to detect viruses. We need amplification. We need better ways to clean the nucleic acid up. So this was a specific target capture, specific target sequence here, and a poly A tail for capture with a poly DT bead. What was nice about it is we could configure it such that once the sample was concentrated on the mag beads, we could do all the washes, the mag separations, et cetera, in that single tube. And then what leads out the end here was to amplification. And the first amplification system was transcript mediating amplification. This was driven by a guy by the name of Dan Cation. We got the technology out of Stanford, but Dan made huge improvements to it. So it uses T7. You're going to see T7 over and over. You're going to see T7 when I get into the CRISPR at the end and the RNA synthesis. So T7 amplification, very, very versatile and useful amplification system. So layer that in, we can now start amplifying stuff up like millions and billions of fold. Layer in something else with the light-off reactions with acronym esters. What happened? So we had Norm Nelson together with others those same guys, Ian Weeks and Stu Woodhead. Stu and Ian were consultants to GenProbe. They were the acronym master experts. We could use a flasher and a glower and discriminate Neisseria gonorrhea and Neisseria meningitis in the same assay at the same time. These are Aptima Combo 2 assays, is what it led to. So add that in, add in a real-time version. This is exactly the same as TMA, but runs in reverse, uses a blocker. It's just like TMA, but it runs, it's a reverse TMA. GenProbe uses it for real-time. And so you take specific target capture, this reverse version of TMA, and those molecular torches that Mick Becker invented, and you can do real-time also. So this isn't CT numbers, this is just time. So as amplification takes place, it self-reports. So this just shows the output of a prototype PCA3. PCA3 is prostate cancer assay. Ended up not getting moved into the PCA product, but again, very useful. So this is the repertoire that, starting with that core of the onion in the very beginning, putting layers on, of just building it up. So HPA, I should mention all of these culture confirmation assays still bought and sold today. And then you layer in there was a MagSep, then the hybridization protection assay plus TMA. That was the MTD assays for detecting tuberculosis, and then chlamydia, and then this whole series of Aptima, target capture, TMA, HPA, and the DKA, the dual kinetic assay I mentioned. Whole series of assays using that IP. Get down towards the bottom, we have a target capture, reverse TMA, or real-time TMA and torches, and then on the Panther fusion they now have with Hologic since the acquisition of GenProbe, they have universal target capture, PCR, and TACMan. The one very cool thing I will say about the Panther system, and it was kind of nearing the end of its development when I was at GenProbe the second time, is that the Panther runs all of these assays on it on a single platform. So it will do endpoint, it'll do TMA and endpoint chemiluminescence. It will do real-time molecular torch fluorescent real-time. And in what we call the sidecar, that's where the fusion part comes in, it will do universal target capture. Universal target capture is very similar to target capture, but it captures everything. It's still that first hybridization and a second tailed hybridization. It will also do now universal target capture, PCR, and TACMan. So all three of those out of the same instrument. The target capture up front is common, and then it splits streams depending what you want to run. So that's kind of the GenProbe story. This is the Panther fusion with the TMA Panther version and the fusion version. And again, I don't have any vested interest here. I don't own any stock in Hologic, et cetera, but just trying to give you guys insight in terms of how these things can be used. The other thing I should mention about that Panther fusion is it's an open access. You can build your own assays and run them on that instrument as you like. And coincidentally, when I was at GenProbe second time, we also got the National Medal of Technology Innovation presented by President Bush at the time to Hank Nordoff, who was the CEO at the time. It was a nice recognition of what the company had done. So I'm going to give you one other example of molecular diagnostics, but with a whole different concept. This is using a thermodynamic trick to very precisely recognize very rare mutations of interest. So this is a story around using an analog assay, not digital PCR, to detect rare mutations very sensitively. And with an analog assay, a problem that most people have is discriminating a mutant from wild type. That if you try to go in and selectively amplify a mutant, you coincidentally get some background amplification of the wild type or breakthrough amplification. It can also be amplification errors. There can be deamination of cytosines, which makes a cytosine look like a uridine, and polymerases looks like a T, so you get a C to T transversion, et cetera. So this assay takes into consideration all those challenges, especially the suppression of the wild type. So here's the thermodynamic trick. And this is, again, sort of a what, why, and how. What did we want to do? We wanted to detect mutations very, very sensitively. Why? So we could detect rare genetic events out of circulating tumor DNA. And so this was the how. The how was to use these switch blockers. And the way that they work is that this anchoring portion of that switch blocker, if you imagine the top of this table, is 3 billion base pairs. That anchor brings that switch blocker very specifically to that one little, basically fly speck, where you want the blocking to take place. So first it anchors you where you want the blocking to occur, but the other key thing it does, increases the local concentration of that shorter, much higher specificity switch region a few thousand-fold. So now the switch will hybridize when it would never hybridize before. So it will stay, if it's a perfect match in that switch. Here's an example of a single nucleotide change. This is associated with T790M. It's a G to A transversion associated with the resistance marker in the EGFR pathway, which is very pertinent to people with lung cancer. But this is just what's showing here is the performance of that switch. That if you imagine this is the melt curve when there's a mutant there of the switch opening up and the quencher now quenching the fluorescence. So this is a derivative, so you can see how the melt curves are. Right here is about where the switch is starting to open pretty well with the mutant. It's all the way up here before it starts opening with the wild type, 20 degrees away. So 20-degree discrimination. This is fairly unheard of due to a single nucleotide change. This also reports in real time. So here's again how it works is that it's everything that I just mentioned, but that when it's a wild type, switch is tightly closed because of a perfect complement. But now these switches are like 7, 8, 10, 13, et cetera, very small. So you're seeing small differences in a short switch region, but this now switch, because it's now been recruited by the anchor portion, has an intrinsic TM into the low 70s. So wild type, tightly closed switch, raise the temperature up a little bit, it's still closed, blocks the forward extension of the primer for PCR. There's a mutant in there, switch opens readily. When it's open, it can extends into that bridging region. I didn't mention the bridging region. This basically holds things in register. It doesn't hybridize. We have an anchor, a bridging sequence. So it's like the anchor is like your forearm, that bridging region's like your wrist, and the switch is like the palm of your hand. Perfect match, switch tightly closed. Mismatch, switch opens, amplification happens. So this is the performance in real time due to a single nucleotide change of when the mutation is present, switch open, amplification happens, it's reporting in real time, 52 degrees to 72 degrees, good to excellent amplification. Wild type, little background drift, nothing. Doesn't care that wild type is there. So we're getting at least a 20-degree discrimination shown here. If you compare it to a normal deoxy oligonucleotide, the TM discrimination of a single nucleotide change in a long sequence that would have a TM in the, say, 73-degree range, something on the order of a 30 mer, about 0.7 degrees using nearest neighbor calculations. We're getting 20 degrees. So we can multiplex these things. These can have slightly different amplification profiles across a number of mismatched targets and easily discriminate from wild type. give you an idea of how good the performance is, this red line is a dilution series of the mutant. 7,000 copies, 700 copies, 70 copies, 7 copies. It looks theoretical, that's the actual data. But it's not only the actual data of a dilution series of detecting a mutant down to 7 copies, it's in 42,000 copies of wild type. So basically the assay doesn't care the wild type is there. These are cartoons that shows what the curves would look like with 1%, 420 copies, 42 copies, and 0.01 or 4.2 copies of the wild type. So suppressing wild type very, very effectively. What's this translate to and how do we use it? So we can take a crude minor allele frequency detecting instrument like a Sanger, put this on the front end, and what you're seeing here is that same T790M, but it's 14,000 copies of mutant of wild type in 7 copies of the mutant. So it's 99.95% wild type, and that's 0.05% mutant. There's not a hint, if you're no selector, no selective blocking, not a hint of seeing that 0.01% mutant in that 99.95% wild type. But if we do the selective blocking, now all we see is the mutant A. The 0.05% mutant has been so selectively amplified up, there's not a hint of the wild type that was there initially at 99.95%. So you also couple this with next-gen sequencing. We are reducing the background of next-gen 100 to 1,000-fold. Most of what you sequence when you sequence with next-gen is wild type you don't care about. So we use these assays on the front end next-gen, suppress the wild type, amplify up the mutants. So at Biocept, this has been used to clinically validate a whole series of assays that are offered clinically for patient decision-making purposes and comprises these assays for lung cancer and colon cancer, melanoma, et cetera. Detection off ctDNA with real-life patient samples with reports going out to their physicians to aid medical decision-making, tell them what drugs to be put on as what pathways affected. I'm going to give one super brief example of therapeutic. This is the next story, but it's very brief. So a company we spun off of GenProbe was called Genta. Genetic Therapeutic Applications is sort of what it supposedly stood for. And we got rights to methylphosphonates for antisense and phosphorothioates, et cetera. But at Genta, we were very keen on further increasing the potency. So this is, again, a what, why, and how of what we wanted to do with antisense oligos. We want to make more nuclease resistant. We wanted to get them so that they could be more easily delivered, and we wanted to get their TMs up so they're much more effective antisense molecules. So this is a little vignette. This is the most potent antisense molecules that we came up with. Didn't take them into the clinic because we had to move faster into the clinic. We went with phosphorothioates, but we had looked at, because we had rights to the methylphosphonates, these came up with a construct. Again, I was talking about methylphosphonates providing nuclease resistance to 'O-methyl, increasing affinity. Well, here we put them together. But methylphosphonates also had 2 chiral forms, an RP form and SP form. The RP form had favorable properties. The SP form, not so much because it destabilized hybridization. So came up with these dimers that were chirally pure methylphosphonates mixed with diesters. They were easier to deliver. And then the 2'-O-methyl was put in to give more nuclease resistance and increase affinity. This is a paper that we published on properties starting with racemic methylphosphonates work really lousy. This is more thermodynamics than probably most people in this room want to look at, but the reference was on the previous slide and on the next slide if you want to dive into it. But we essentially took a methylphosphonate, a TM at 34 degrees, And by then making it 2'-O-methyl and then chirally pure methylphosphonate interchanges, we took that 34 degree TM to 70 degrees. And when we looked at these, they were bulletproof in terms of stability compared to even 2'-O-methyls that are over here. Here's again this reference. This is back in the 90s. Patents were issued on this and have just recently expired. So I think it's something that people might want to reinvestigate, or you use in some other different way. You want something that's bulletproof, has high TMs, bulletproof to nucleases. This is a construct. It was the most powerful construct we looked at in an 8-year time frame of working on these antisense oligos at Genta. So now, the final story. New story. This is synthetic RNA. So, as I think everybody in the audience probably knows, RNA as the template for making protein has a 5' cap and has a poly A tail. Those are, number one, the 5' caps for recognition by the translation systems, but also to provide nuclease resistance so things don't get eaten up from the 5' end. And a poly A tail that also gives you temporary nuclease resistance until the tail gets eaten so far down that the RNA is not active any longer. So we're back to T7. I told you T7, this was the basis. T7 promoter was the basis of transcript-mediated amplification and real-time transcript-mediated amplification. Very versatile. It's also used at TriLink to make, on a custom basis, for any of you that are interested in the audience, your own personal, for whatever you want to use it for, messenger RNAs. So what TriLink has come up with are these cap structures that are recruited by the T7 polymerase during RNA synthesis. It's actually pretty amazing. What happens here is that these dimers bind better to the T7 polymerase and the template than a monomer. So if you put those in, they get incorporated preferentially at the first 2 to 3 nucleotide positions. So instead of normal monomers going in as the triphosphates, these things go in and first dock and then get added onto. So they get grafted in during T7 polymerization. And in TriLink's hands, this capping efficiency, using this kind of method, good old T7 polymerase, 90 to 99% efficiency. And they can graft in a lot of other targets other caps as you like. They also can construct using plasmids targets that already have that T7 promoter in, as well as that poly A tail. And so RNA of your choice, they'll have this plasmid constructed that the poly A tail automatically has 80 poly A's into it when it gets translated, but that, I should say transcribed. But if you want them longer or shorter, they can do that. Much the same technology is used to affect gene editing. So now with this ability to make RNAs in hand, TriLink makes and provides to whoever's interested the RNA to inside cells make Cas9. So pretty amazing thing if you think about it. You take this synthetically made RNA, transfect it into a cell. It is basically recruited by the protein synthesis machinery. It'll make Cas9. There's also these guide sequences that are needed to drive the Cas9, And if you want to do a homologous recombination, a single strand. All those things can be combined into your favorite transfection reagent, introduced into cells, and edit as you like. Additionally, with the single guide RNA, if you want them to last longer, remember in the beginning talking about those modifications on the 3' and 5' end to protect against nucleases. You can graft in, or you can incorporate in thioates and 2' methyl and give them more nuclease resistance, and it'll work even better. So this is, again, how these work with the Cas9, the guide sequence. If there's no single-stranded template put in to drive recombination, there'll be a few nucleotides clipped out of here. It will cause a frame shift and activate the gene, or if you want to do a gene repair or insertion, you provide, and this can come from TriLink, a single-strand DNA template to drive that recombination. So at this point, sort of my goal has been sort of give you a good idea of the different ways that you can use modified oligonucleotides to help with your own what's and whys, maybe a little bit of the how. So sort of the take-home message at this point is if you successfully utilize modified oligonucleotides, you can avoid wrong turns and dead ends, can help separate you from the crowd, and allow you to drive great innovation for a bright future ahead. Now, sort of last final 2 slides, this is kind of like an eye chart, but a lot of people contributed to the core technologies that I've sort of gone over today. I'd mentioned Jerry Ruth already, and all of the base modifications that he did that allowed us to decorate nucleotides at various locations. I was one of the co-founders, together with these 4 guys, Ken Witter, Vince Frank, Drew Ye, and Rick Tullis, what was the 3rd biotech company in San Diego. Now there's hundreds. At GenProbe, for me, company number 2, Dave Cohn, the guy that came up with the ribosomal RNA target idea. He was also a co-founder of the company together with Tom Adams and Howard Berndorf. And then contributing to the cores of technology that drove all of the initial GenProbe assays, Norm Nelson, Mark Reynolds, Ron Bott, and Alex Waldrop across various technologies. And then more advanced technologies as we added the onion, and I joined GenProbe the 2nd time. Dan Cation and Tim Foltz, TMA, Mick Becker on lots of different inventions here, including the RTMA and the reverse target capture, numerous others. As far as Genta, that was Mark Reynolds, Rick Hogarty, Morty Vahefi, and Terry Beck. Those are the 3 guys that are TriLink, or were TriLink, or were the initial founders. They had been part of the R&D team at Genta and ended up becoming a TriLink. Bob Brown, Tim Riley, co-founders with me at Oasis. I didn't have time to go into that Antisense library stuff. GeoBiotechnologies, the switch blocker stuff, Natasha Paul worked with me for the initial reduction to practice of the switch blocker technology. Norm Nelson, who goes all the way back up here, so I've worked with Norm for a lot of different years. He's a co-inventor with me on a portion of 7 different issued patents on target capture, amplification, next-gen sequencing, library construction at AGEA, and then a number of people at Biocept that drove into the clinic the switch blocker technology. So I want to say thank you at this point. These are my contact email addresses at both AGEA and Biocept. Feel free to contact me. Happy to work with you in collaborative ways, driving technology, or in other ways at either company. Again, GIA is primarily a technology core company, Biocept, a liquid biopsy company. I put this figure in here. It's just kind of a reminder again, DNA is a right-handed helix. So if you imagine shovel stuck in the ground, handle sticking up, you grab it with your right hand, the helix turns in this right-handed direction. If you grab it with your left hand, it's the opposite direction. So when you go throughout the meeting in the next few days, just keep an eye out for how often people get right-handed helix correct versus left-handed. I think artists like left-handed, you'll see it as error. And if you even look at this brochure, very top cover, don't need to go further than that. So again, thank you very much for your attention. I hope it helped a little bit with what you can do with oligos for a bright future. Thanks.